Hi guys, I just wanted to give you an update of what we've got going on in the lab and give you a thumbnail sketch of how the connections between some of the resources that you've been able to provide us have been able to help drive the current collaborations that we have, and then what we have looking ahead in terms of these different efforts and how they're going to come back to help our university and our lab as well. So our focus is looking at Parkinson's disease and different strategies for treatments and cures, basically trying to protect or make new dopamine neurons. And so a lot of this arose from the looking at uh, different, looking at a mechanism of action in terms of how our particular gene of worked. So what this is illustrating is basically it's a graphic where we're placing our translational neuroscience lab, which is essentially what our lab is developed into. And we started with looking at sort of how this particular gene NATO3 is important in how it functions and what's its mechanism of action in the developing embryo. And so that was what we had been chipping away at for a long time. And most recently, we've been supported through this Campbell Foundation grant that'll be wrapping up in July. So we'll be continuing that effort and looking to see how it is that our gene works in the developing embryo. And the important part about it is that that gene's really helpful in terms of driving other types of factors that are important for making dopamine neurons, like we see over here, or protecting dopamine neurons, like we see over here. So in a sense, our efforts that we've been working on for a while have now been able to sort of blossom in this petal diagram of a flower here into basically supporting neurogenesis, helping to drive and make new dopamine neurons, and then also neuroprotection, helping to protect dopamine neurons that we have. So I'll talk a little bit about that science in a bit. But these are supported by different grants that we have currently. The Advanced and Commercialization Grant is in this space here, where we have the intellectual protection that we've been able to work through with the CSCE and the TCO have been really helpful in terms of us being able um, to have access and be eligible for funding that's linked to pursuing this question of basically the gene that we've been characterizing here, when we modified it, can we use it to make new dopamine neurons? And then over on this side, we've been supported by the NIH is really excited about our work and supported a collaboration with us and Van Andel to basically test and see whether our modified gene would be important for neuroprotection, protecting dopamine neurons from destruction. So those are the different types of petals that we have going in here. These two petals are ones where I can see potential for growth. Down here is for Spectrum Health. There's some funding mechanisms that I've recently learned about that we may be able to use to help funding for collaboration between different institutions. So I'm looking into that. And then MSU, in addition to Van Andel, has a lot of pretty active types of folks, and some that I've been talking with to see whether they might be able to help out some of the efforts that we have. And I think this is really kind of an interesting space because these individuals who come from here are ones that they have a lot of training and they can be helpful in potentially recruiting into different departments. Like we have two affiliate or adjunct faculty in chemistry and biomedical science who came from labs that were involved in this type of work. So pretty interesting type of area for growth. But we're going to be focusing on these upper three petals here. And let me just kind of tell you a little bit about the science that we have here for Parkinson's disease and how this is, and where we fit in it to give you a bit of an idea of how it all works. So this is depicting sort of number of dopamine neurons that we have and over time. And so as we kind of move down, we have a decreased number of those neurons available. And so we're born with a full complement. And then the idea is that we basically lose those cells over time. This happens with normal aging, but in Parkinson's, it's accelerated. And so that you end up progressing from being pre-symptomatic all the way to having partial and classic motor symptoms here. And so the clinical rating starts to increase as the number of dopamine neurons drops over time. So our goal basically is to try to see if we can actually either protect the neurons in these stages, or replace neurons that are lost in this stage here. And what's really interesting is that our gene actually, that our modified gene that we have is one that we can actually be able to characterize. So that middle petal that I showed you in this graphic, if I can go back, there we are, here, this mechanism of action, I'll tell you just briefly about how that worked, but and link it to what we were looking at ahead, assuming that the machine works with me. Good, here we are. And so what we're looking at basically is like what we've been doing in the past, and what we currently are working on now is that we've been using these injecting DNA into the developing embryo. These are chick embryos, and then we're able to monitor and see what happens to genes that have been expressed in different areas of those chick embryos. And so this glowing green area here is indicating cells that are expressing our gene of interest. And this little bulb is just is a midbrain where dopamine neurons are actually born. And so we can actually go and inspect using anatomical techniques and look at these different cells and then basically interrogate them and see, like, do they look like dopamine neurons or do they not? And so on. And that's been helpful in us being able to get to where we are with our analysis. And then over the course of my sabbatical, we've been able to kind of bring this into cells and different types of cells to be able to ask questions about how this works and the mechanism of action of how this particular gene has been able to exert its effects in the developing embryo. So, so that's a bit of that middle petal there, and that's been all undergraduate students. One graduate student so far has been engaged in that process. So. What's been interesting is that when we've modified our genome, as we kind of took some of our insights that we got from these studies here, we've been able to find that it's relevant for how we actually make dopamine neurons. So the factors that help to drive dopamine neurogenesis, these are genes that that are important in driving dopamine neurogenesis. So if you add them, sometimes in combination or sometimes even just by themselves, well, usually with other factors, you have to have them when you put them into cells, they can help promote and create dopamine neurons. And so this idea here is, here's a test tube. These are little particles of mechanisms in which we can actually make these genes, introduce them into cells, and then they turn into dopamine neurons that we can put into people's brains to help replace the ones that are missing. So they have strategies like this for in phase 1 clinical trials, both here in the U.S. and in Japan, using different iterations of that strategy. And then there's also the potential of actually being able to help reprogram resident cells within the brain as well, which is pretty new, but is out there as well. But the tricky part is, like, it's just a combination of these different factors here, and it's a lot of different things to coordinate. And what's interesting is that our gene helps to drive a lot of these different factors that are important for making dopamine neurons. So what we'd like to do is basically be able to see whether our gene can actually drive this human embryonic stem cells and provide maybe a simpler or easier way to help create dopamine neurons that we can put into and use for therapeutic treatment or for research analysis as well, and characterizing dopamine neurons that we can create from cells that people have on the like fibroblasts and so on. So that's a collaboration that we have going on with the Human Embryonic Gene Editing Core. So formally, they're actually we're subcontracting with them to work on characterizing this gene and see whether it has this capacity or not. And so we have some promising results on that in terms of some of these markers are going up when we actually put in our gene. And so that is in progress. And so we'll be taking those results and then applying for more funding, which I'll talk about in a little bit. So this other petal that we have here, neuroprotection, let's dig in on that. Now that we've talked about neurogenesis. And so these same suite of genes that we were talking about before for making neurons are actually important for protecting adult dopamine neurons. So, one context we were saying, we wanted to change stem cells into dopamine neurons. Here we're saying if we drive these different expression of these genes in dopamine neurons that are already there, that can actually help them survive different types of challenges that can come up in Parkinson's disease. So these neuroprotective and survival pathways are activated. And that makes it so that PM-Theta3 might actually be a really good candidate for being able to support and help those neurons. So so the NIH was excited about that as well. So when we applied for it, so we got funded, and our aim that our lab is involved in, and that we're collaborating deeply with Van Andel on, is basically looking to see if we can take human dopamine neurons. So these aren't human embryonic stem cells; they're just a cell line that's derived that are human dopamine neurons, and then basically putting in our phosphomimetic NATO3 into those into those human neurons. This is our modified gene that we've been playing with, and seeing if we can upregulate those genes and protect them from the conditions that mimic Parkinson's disease. So that kind of outlines these different petals that we have here. And then so at the center here, then we have our neuroscience lab. We have a postdoc in the lab right now, Dolly Chowdhury, who's been really helpful in us being able to pursue and support the commercialization components here, and more broadly, be able to help actually support all three of these different petals through her efforts. So what's nice is that, her actions and her activity is basically helpful in not only keeping the lab running and efficient and effective, and provide a nice professional face for our lab, in addition to myself, of course, with both Van Andel and U of M, but also be able to help develop the culture of the lab and training opportunities for students as well. So she's been an essential component in the success that we've had so far. So let's talk about how resources and plans ahead might be useful. I mean, we've talked about a little bit of the science here, the neurogenesis, neuroprotection, and the mechanism of action. And I should say, this a lot of this work has involved students from Grand Valley, and undergraduate students have been key components of that work. So really student-focused type of efforts and outcomes there. This also helps us really, I think, connect to the health science corridor that we have within the state. I'm not just talking about Grand Valley and Grand Rapids, but just the types of collaborations and the quality of the collaborations that we have are putting us on the map in that kind of context. So we have, for example, the guy who came into there's an individual I invited in for the biomedical science seminar speakers series, and he was a reviewer, or is a reviewer, for these NIHR-15s. And so he saw our university, he saw the work that we were doing, and those two, the university infrastructure, the support that they have for students, engagement of students, he was saying, you really have to send your grant to my committee. I really think this is a good position to be in. So that's really exciting, because that helps illustrate how much of the support that we've enjoyed from the university is really connecting to opportunities that we're going to pursue, and then also that we have this. So he's from Wayne State, then we have University of Michigan, we have Van Andel. So we've got basically all ends of the state involved so far. So I anticipate that will expand, and that will be this global phenomenon. But let's focus on what we have kind of coming up in terms of our plans here, in terms of those different petals. So that cellular mechanism, that top petal that we were talking about there, we're currently supported with a Campbell grant that ends in July. Then we'll have we'll be applying for an R15 in February. I'll show you a map, kind of how we're looking at this aspect of it. But I'll be asking for a course release at that point. Neurogenesis here, we're currently supported with Advance and the T3N support there as well. So that ends in March. We'll be applying for a Kickstarter grant and an MTRAC. So there's two match components there. But I think this really helps us to be able to realize the commercial potential out of that. And then there's these tech transfer opportunities that can come up down the line. These are the indirects that are potentially available with that. I'll go through some of the details with that, so you have a realistic appraisal of what those opportunities are. But we're looking at 2021, 22 on that. And then for neuroprotection, then what we have here is that's at R21. I'll be, if the data looks interesting in a way that we can actually be able to help kind of develop it into an R01, then we will. And so we'll have to see how the R01 and the R15 articulate with each other. You can't have an R01 if you have an R15. So, but those things, we can work them out. The data's pretty early on. We're still putting together a lot of tools that we need to be able to actually put together a paper that's going to have significant impact. So we're that early application in October 2019 is likely going to be later. But I'm going to be applying for a Campbell Foundation grant to help support some of that activity in February. So that has some match that's associated with it of 6.9K. The postdoctoral support that I was describing to you before, it was Dolly Chowdhury, who we have in the lab right now. So this is these are typically renewed for a second year. The 50% of the salary is covered at 20 by the university, so that would be 25,000 there, and then there's 16K for benefits. So so that's the so there's a match component there of 41,000. So Linda's in conversations with the folks there to learn about the shape of that opportunity and the likelihood of us actually being able to get that kind of support. But I think the access to be able to that match is important. So I put a range here of 21 to 62,000 in terms of these match requests, because I'm just, we're learning about the status of this currently. So I wanted to give you an idea. Okay, this is some of the ask that we're thinking about here, but I think it kind of, you can see how it links to some of these really exciting advances that we have, and how that's important in therapeutically potentially, and having students involved as well. And I wanted to kind of just end this show for a second and show you some other stuff that we've been kind of working on. Give you an idea of the some of the different grants that we're looking at and pursuing. So I've described to you, sort of shown in the gray boxes are the names of the different grants here. And then this is timelines. And so just kind of giving you an idea of what we're considering. I'll include this in here, so just for just in case that's helpful for you. But T3N is currently what we're using to help support the postdoc that we have in the lab. And so this illustrates the amount that we were given, then the match that we had to come up with. And so because we had the benefits in addition to the covering 50% of that grant, we come up with 41. So that grant total was 66. We'd be looking to apply for that potentially sometime in early 2019 to help renew that. So that would be potentially a match of 41. And then this is the advance that we currently have and in play. This is the Kickstarter grant that we would be applying for. So these two grants, I think we can apply for and get. So Linda Chamberlain has been able to work with the tech transfer community that's responsible for these types of commercialization linked grants that we have. And basically as for non-R1 institutions, we've been able to have it so it's actually 10% of the total grant amount. So these are the match requests that would be coming up in 2019 for those. So let me talk a little bit about these tech transfer components here. So these two phases, I think, are, so I did the NSF I-Core program, which looks at sort of helping to develop the commercialization potential. It's actually been very helpful in terms of writing up the grants that we have for the advance and T3N and so on, sort of understanding the language and the needs that are connected to that. So these tech transfer grants that we have here, these STTRs, they come, there's two different phases. I think out of I-Core, we may be able to apply to something that's sort of a combined 1 and 2 phase here that combines the two grants, but we may have to stick with it sequentially, but these will be helping to help define the markets and then the R&D to be able to help support this effort that we have and be able to bring that PM803 into something that's useful technically and can be used in a commercial context. So we've been talking to Cell Dynamics International. My collaborators at Van Andel at Rush University have connections there. And so that's the potential commercial partner that we could be looking at down the line. And that helps us really be able to define these different partnerships that we can use to be able to pursue these opportunities. So, so I'd be asking for course release as part of this quote unquote match to help manage those grants. And then these are the, I estimated just 41% on our indirect, so you have kind of an idea of what would be coming through. So these are in thousands, so this is $1 million grant here. And so those are the indirects that would be coming through. The phase 2B stage here, this is a little more of a, there's a lot of things that need to line up for that component to work. We need to make sure we actually have our commercial person lined up and that there's significant enthusiasm in terms of the way that the product works and it would actually be able to help. So we really, these two phases will help us define whether we get to this phase or whether we, our product's really not where the state of the R&D is. So a lot of the research and development that's happening here will help determine whether we can actually get to this point. But if so, then it's a $3 million grant and a healthy indirects. The R15 that I'll be applying for in February is over here. So there's indirects that are associated with that, 300,000 over 3 years. And then it's renewable, so we'll be applying again, presuming that we get it the first round. And so we can see that kind of happen here. And then the Campbell grant, which I've listed here, is supporting mechanism of action. In the slide I was showing it was supporting this neuroprotection type of work here, but we'll be applying for support. And that has a match of 6.9. So, so that gives you an idea of just, I think I wanted to kind of illustrate that we're really careful and thoughtful with the stewards of the support that you guys have been giving us, and that we're really thinking about how we can get these things launched into exciting opportunities that are great for the university, the students, and obviously the efforts that we have in the lab as well. So I hope this has been useful, and that I didn't talk too long, 22 minutes. But I think it will be useful. So thanks, and I'll look forward to hearing from you soon. Take care.